Yes, guys. Good evening. Good evening, all. Yes, Dikshit, Shraddha, Nila, everyone. Good evening. Hope the audio and the video is clear for you. Today we are starting with a new chapter that is Cell, the Unit of Life, and the first lecture from Point Me. Yes, guys. Okay, fine. Only five. Everyone hit the like button. Yes, we are moving on to today's lecture on Cell, the Unit of Life. Okay. Okay, we are moving on to the chapter cell. So everyone have heard of this term. Everyone have studied this in the ninth standard. What is cell and what are its function? What are cell organelles? All those things you might have learned, right? So what is a cell? A cell is the structural and functional unit of all living organism, right? So the structural and functional unit of all living organism is what we say to be a cell. It forms the basis of life. Okay, the very minute component of our human body is what? Cell, comprising of what cells which combine together to form tissues, which combine together to form the organ system, and to a human being. Okay, not to a human being, to a organism, right? So that is what we had learned about cell, and we have learned about its organelles, prokaryotic, eukaryotic cell theory. That's all what we are going to discuss here also today. Okay. Yes, good evening, Pradeepa. And now we have who discovered the cell, from which cells which were discovered, right? Yes, Roshita, you're back. Good. You are a neat aspirant or 10th standard? Because in the previous lecture also you were there. Yes, Shivani, good evening. Yes. So if a question is asked as who discovered cell, a cell is discovered by a scientist known as what? Robert Hooke. So who discovered cell? Robert Hooke. But who first discovered a living cell? If a question is asked like that, who first discovered a living cell? Okay, Roshita. Who first discovered a living cell? It is Anton van Leeuwenhoek. Clear with that? So first, Anton van Leeuwenhoek discovered a bacterial cell. He observed a bacterial cell and discovered that. And if a question is asked, is who discovered cell? It is answer is Robert Hooke. And Robert Hooke discovered the cork cell, observed the structure of cells from the cork cells, and what concluded it as a structure like honeycomb, right? So these are the what some of the histories of cell discovery, right? So what is cork? So from which plant is this cork taken? The cork cells are taken from which plant? Anyone know? It is taken from a plant known as curus. Oh my god, it's very difficult for me to write. Okay, curus, or we can say it as the oak plant. Oak. Yes, no issues. No issues, Rajni Bal Chituri. Harshida. Okay, fine. Srujana, okay. Everyone. So these cork cells were obtained from the oak tree, also known as curus. And the invention of compound electron microscope revealed the structural details of the cell. And in later, Matthias Jacob Schilden and Theodor Schwann formulated the cell theory. So according to your concept, who proposed the cell theory, guys? Comment me fast. Who proposed the cell theory? Fast, waiting for your answers. Children and Schwann. Okay, but most of we have heard of one more other another one more name known as what Virchow, right? Rudolf Virchow. So we should know why he is omitted from this what. First, when we say about the cell theory and all, why he is omitted, right? So all these are the things which we want to find, and exactly Shraddha Virchow expanded the cell theory. That is, he proposed a modified form or the modern form of the cell theory that we will be dealing at the further slides. Clear? So, Matthias Jacob Schilden or Schilden was a botanist. He was a botanist. And this Theodor Schwann was a zoologist. Yes, Dikshit, pre-existing cells. And this, oh my god, it's not getting, okay. And this Theodor Schwann is a zoologist. So, in NEET, a question is asked, okay? In NEET exam, the question is asked, who formulated the cell theory based on the plants, based on the plant features, okay? No, Harshida, we can say Virchow proposed because he added one more point by modifying it. By modifying me means that we added one more point. So we can say that we can say what Virchow proposed cell theory because one more point is added by him. Contribution was there, right? Yes, German botanist and British zoologist Matthias Jacob Schilden and Theodor Schwann formulated the cell theory, and Matthias Jacob Schilden observed that the plants are composed of cells, okay, and Theodor Schwann reported that the cells had a thin outer layer of plasma membrane. He also found that the plant cells have a cell wall, and he stated that the animals and plants are composed of cells. This is the first point that they stated, first postulate of the cell theory. All living organisms, that is both whether plant or animal, it is composed of a cell. Clear? So this was the first postulate that they discovered, and the second postulate that they contributed was that all activities present in an organism is some or product of their cells. Any of the metabolic activities occurring within a cell or occurring within an organism is the sum or product of the cells present in of the number of cells present in that organism. Right? Clear with that? Everyone give me thumbs up if you are okay with this what I'm teaching. If I'm moving fast, you can say that. If I'm moving slow, you can say that. Yes, okay, fine. So this is what we have to say about the first cell theory or what was the basics of cell theory. And on moving, we can see that in 1838 and 1839 was this what Jacob Schilden and the other one Theodor Schwann proposed all this cell theory, right? So yes, Harshida is ready with all the things that I want to explain, right? Yes. Okay, fine. So Harshida just typed and keep it with her. Till then, I just move on to the next topic, right? That's what I to do in my classes. Okay. So in after that, later, only in 1855, Rudolf Virchow came. Rudolf Virchow came in 1855, and he explained that new cells are formed from pre-existing cells. That's what he contributed. He just said omnis cellula e cellula, right? Virchow just said omnis cellula e cellula, and that's what came with the new modified cell theory. Okay, including a further new point, all cells arise from pre-existing cells, which is something contributing towards the reproduction of cell. Okay, we said the composition of an organism. First, what Sheldon and Schwann just said that about the composition of a cell, that it is composed of all organisms are composed of cells, and the prod and the activities present in an organism is the sum and product of their cells, right? And after some years, Virchow came and said omnicella e cella, and that's what gave rise to the modified cell theory or the modern cell theory, including all the three statements. Clear with that? And all these three postulates together contribute the modern cell theory. Okay, now we are just moving on to have a glance on the structure of these cells. Cells may be of two types: plant cell, animal cell, prokaryotic cell, eukaryotic cell, unicellular, multicellular. All these are the classification just based on the cell. So, what is a cell? We should know, right? So, what are the components of a cell? On the basis of what it is classified. So, just we are going to have an overview of this cell. A cell contains a cytoplasm, right? It is a jelly-like, a semi-fluid matrix where all the organelles are placed inside the cell. And this cytoplasm keeps the cell in a living state. This cytoplasm keeps the cell in a living state. Okay. Next is the first organelle that we are going to discuss, the ribosome, which is a non-membranous. Very important, just note it. Ribosome is non-membranous organelle found in the cytoplasm, chloroplast, mitochondria, and on ref endoplasmic reticulum. We can see the ribosomes on all these structures: cytoplasm, chloroplast, mitochondria, ref endoplasmic reticulum. All these structure we can experience. All these organelles we can experience this ribosome. Next, we are going to have the size of a cell. Next topic. Okay, next topic, subheading, size of the cell. So the size of the cell is one thing that varies from organism to organism. All the 10 present, all the 10, hit the like button. So now we have to discuss what are the smallest things, what are the largest cell, what is the size, what is the all those things that we are going to discuss. All these are just useless things. But for need examination, the smallest cell was asked. That is, the mycoplasma is referred to as the smallest cell, that is about zero point 0.3 micrometer, right? Zero point 0.3 micrometer. And it is the largest, which is the largest isolated single cell. It was one of the previous year need question. It is the egg of ostrich, which is the longest cell, nerve cell. But what is the size of bacteria? 3 to 5 meter exactly. What if it us what is it varies from bacteria to bacteria, but an average view of their sizes. Oh my god, what is happening to this mouse? 3.5 micrometer. Human RBCs are the smallest cells in human body. Smallest cells in human body is human RBCs. Okay, and I'm giving you two homeworks were there not there till now for a very long time, so you should find the name of the smallest and the largest bacteria. Smallest and largest bacteria. Okay, clear with the topic for homework. Hope it's fine and clear. Okay, fine. Next, on the base of the shape of cells, you can be say that dislike, polygonal, columnar, cuboid, thread-like, or irregular. All these are different shape of the cells that we will discuss. Okay, and the cells are of two types mainly. That is the prokaryotic and the eukaryotic cell. Clear? Cells are of two types. There is a prokaryotic and eukaryotic cell. So let's check what are that. Now we are moving on to the first topic, the most important. one of the important topic. Okay. Yes, Harshida. Yes, Theo Margarita is the largest. Just don't say it now. You just know Mycoplasma is not the smallest bacteria, Shraddha. Theo Margarita is the largest. That I agree. But just don't say it. You can comment it at the end of the session. Okay, at the end of the session, you can comment it in the comment box. Okay. So move on. Let's check what is this prokaryotes and what are the eukaryotes. Okay. I'm moving on directly to the topic. First time I'm doing such a plan of teaching. Okay, directly moving on to the topic without having any introduction. Let me check whether it is no smallest. It's not mycoplasma. Just search it after the class. You may search it. So prokaryotic organism means there is no membrane-bound nucleus or cell organelles. Okay, no membrane-bound cell organelles or nucleus. So the term prokaryote, what does this mean? Prokaryote. Prokaryote means what? Primitive nucleus. Right? Prokaryote refers to primitive nucleus. Right? So these organisms do not have a membrane bound nucleus and cell organelles. They include bacteria, blue green algae, mycoplasma or PPL. Mycoplasma also known as PPL. That's a pleuropneumonia like organism which causes pneumonia. Okay. They are generally smaller and multiply rapidly than the eukaryotic cell and they vary in shape and size. For example, in case of bacteria itself, right? Bacillus, cocci, what spirilla, vibrio, collar, all those things, right? So all these are classification on the base of shape. What the next topic is the same, the classification of bacteria on the basis of the shape. Bacillus, cocci, vibrio, spirilla. Bacillus is a rod-shaped bacteria. Coccus is the spherical-shaped bacteria. Vibrio is a comma-shaped bacteria and spirillum is a spiral-shaped bacteria shown in the four diagrams as per in the correct order. Look, Bacillus, Coccus, Vibrio, Spirilla. Clear with that? Next, we have the structures or yeah, some of the structures that is used for the classification of this prokaryotic and eukaryotic organisms. So today's session, we will learn about this prokaryotic cells and the next class we will be moving on to the eukaryotes. Okay, one of the interesting chapter if you listen properly. Clear with that? Okay, so the some of the organelles that we use for the classification are cell envelope, mesosome, chromatophores, nucleoid, flagella, pili and fimbriae, ribosomes, inclusion bodies. Okay, take a screenshot. Take a screenshot. I'm waiting for that. Okay, cell envelope. So cell envelope, if we say for the method of classification, in case of prokaryotic cell, we can classify the modification of the cell envelope. Please wait one second. Okay, we can classify the modifications of the cell envelope into three: glycocalyx, cell wall, and plasma membrane. Glycocalyx, cell wall, and plasma membrane are the three basic classification of this cell envelope. So glycocalyx, also known as the cell coat. Okay, cell wall, all of you know. Then plasma membrane, also known as cell membrane, or the latest name, biomembrane, also known as biomembrane. Clear? So, what is the cell envelope? It is a chemically complex protective covering of a cell. It's a complex protective covering of a cell, and it consists of three bound layers: glycocalyx, cell wall, cell, plasma membrane. But in usual case, all these three will not be present in one organism. Clear? Because glycocalyx further have two structures known as a slime layer and a capsule that we will discuss. Okay, surely we will discuss. Yes, glycocalyx is the outer layer. What it is composition is thick. Composition and thickness vary in different bacteria. It may be a slime layer or capsule. Slime layer or loose sheath, and capsule or thick and tough. So glycocalyx, basically it's made of certain structures known as polysaccharides. Okay, basically the composition of glycocalyx, it's made of polysaccharide, and this glycocalyx has two layers: a thin layer known as slime layer and a thick layer known as capsule. So what is the function of this slime layer? Its function is to prevent water and nutrient loss from the organism. Prevent water and nutrient loss from the organism. In case of capsule, their function is to helps the organism to hide from the host immune system. If the immune system of the bacteria or the prokaryote is what is well exposed, and what's the use of causing diseases? Surely the antibiotics or the antibodies will just defeat that by invading it and just defeat that. So just hide the immune system. That's what the function of this capsule. Clear? Next we have the cell wall, and it is made of peptidoglycan. Cell wall made of peptidoglycan. Peptidoglycan composed of NAM and NAG. N-acetyl muramic acid and N-acetyl glutamic acid. Cell wall is the middle layer, and it is seen in all prokaryotes. The slime layer and capsule that we learned in the classification of what this glycocalyx will not be seen together. Cell wall gives shape to the cell and provides structural support to prevent bacterium from bursting or collapsing. Prevent the bursting or collapsing of the bacterium. So, cell function of cell wall is to give the shape and structural support to the cell of bacteria or the prokaryote. Next is the plasma membrane. That is the third layer. Hope it's clear. If you have any doubt, you can comment me. Okay. So, plasma membrane or cell membrane, and the reason name is the biomembrane. Okay. Not that name, biomembrane. Okay, so it is an inner layer. It is semi-permeable in nature and interacts with the outside. This is structurally similar to that of eukaryote. So this cell plasma membrane is structurally similar to that of eukaryote. Okay, cell membrane. It is one of the question asked for the need examination. Name the universal feature in all living organism. Universal feature in all living organism. Universal feature in all living organism. It is the plasma membrane. Not it. It's a previous year need question. Not need. It was when Ames was separate. Ames question. Okay. Next, we are moving on to the Gram's chaining method. Okay, based on the types of cell envelope and response to Gram's chaining, bacteria are of two types, mainly based on it is mainly based on the composition of cell wall. We can characterize this. That is, it's gram positive and gram negative. Clear? Gram positive and gram negative bacteria. So this is discovered by a scientist known as what Christian Gram. Christian Gram is the scientist who discovered this gram staining method. Okay, one thing happened is that during the time, it's one of the story that occurred when during the time of plague. Once the plague was a widespread disease like this COVID. Okay, once it was a widespread disease and it cannot be controlled. Over 500 crore of people died due to this plague, right? So I think in Sweden, or yeah, in Sweden, there was a what we say in a village. Okay, in a village, all the people, most of them died due to this plague, right? Most of them died due to this plague, and they thought that surely they all will die. The entire society will be lost due to this disease of plague, because no, there were no vaccines, there were no discoveries yet to prevent this disease. So what happened is that they all decided to shift that place. Just what, what we say, just leave that village and move to migration, some sort of migration. Just move on to some other place for a better future, for safety of their children. Maybe it might be like that. Are you all here listening to this story? Okay. So they just decided to move that, and there was a doctor. So it's not at all related to this gram staining. Okay, but anyway, I'm saying that in order to make you aware of how this COVID is also spread in the same way. So if you all are here, please come in me. Okay, fine. Yeah, there was a doctor, and all of them in that village. In past, there were only one doctor in for one village, like that. A single doctor who is having knowledge in everything, like that. They all had a great belief in that doctor, and they seek the doctor and told him, since there is no vaccine till now, we are leaving this place and we are moving on to other. Okay, and what happened is that the doctor advised them not to leave. And they asked why? Why should we stay here and leave our life, spare our life for this what nonsense of such a disease known as plague, right? So, but the doctor said, you all might be certain carriers, might be you all having that disease, but there will be no symptoms because of your high immunity. So if you are moving, shifting from this village to next village, what happens? You will be the carriers, okay, and it causes in the spreading of the disease to that village also. The village which you are gonna migrate also will be affected by this disease. Okay, so since that doctor told like that, they all decided to just not to leave that place, and they just stayed in that village, and they just locked the entire roads and openings of that village. That's what we did in this COVID situation known as the lockdown, right? Right, in the same way, the first lockdown, right, in Sweden due to this plague disease. Okay, so like that they just closed all their village, all that villages openings, and then what living places, everything where roads and pathways were closed, and in order to prevent other villages from affecting with this disease, right? But by that time all the villages got spread this disease, but they did one thing in order to prevent the disease from their village to other village. Like in the same way, what happened is that they all died, including that doctor died affecting this disease. Affecting this disease, that doctor too died. All the citizens in that village died without even a single organism. That's what happened. That's what the effects of this plague at that time. No medicines to cure that. So in the same way, in order to prevent this viruses and or one thing, we should spare our life. I'm not asking you to spare, but we have certain medicines at least for curing this COVID, right? But at that time, there were no medicines to cure this plague, and they all died. No one, sparing no one in that village. So, in order to, for if you affect with a disease, just don't go. Just don't go somewhere. You just stay in your home. You will be cured from that. Okay, do not be a carrier and spread the disease. That's what I said. Okay, the time went for 45 right now. Okay, then gram positive and gram negative bacteria, this is what all about that. Okay, based on the gram staining, it is classified into two: gram positive and gram negative bacteria. Gram positive, they take up and retain the gram stain. They do not retain the gram stain. That means gram positive bacteria, if we pour the stain, it just take absorb the stain and just retain it. But in case of gram negative bacteria, stain doesn't affect. That's how we affect we identify this gram positive and gram negative bacteria. Okay, everyone here. Yes, first lockdown, Harshita. That's what happened. Okay. Then examples for this gram positive and gram negative bacteria: E. coli, Escherichia coli is the gram negative bacteria. Example for gram negative bacteria. Staphylococcus, Staphylococcus aureus. It is a gram-positive bacteria. Staphylococcus aureus is the gram-positive bacteria. These are the examples of gram-positive and gram-negative bacteria. Clear with that? Next, we have the cell organelles, mesosome, and chromatophores. So this mesosomes and the chromatophores and the membranous structures. Mesosome is formed by the infolding of plasma membrane. Look, the infolding of this plasma membrane forms the mesosome, including vesicles, tubules, and certain lamellae. These are the structures present in a mesosome: vesicles, tubules, and lamellae. Okay. So what is this mesosome and what is this function? That's what we have to know, right? What is their function? So their function is that for cell wall formation, for DNA replication, for distribution of chromosomes to daughter cells, for respiration and secretion, to increase surface area of plasma membrane. All these are the functions of mesosome. Guys, if I just do a line like this, you will have this much space. But if I just draw a line like this, okay, if I just what draw a line like this, or if you have a thread like that in this way, first one, this one, the first one, if we have the line number one, thread number one, if we just stretch it, at least just it will expand just a minute also. But if we stretch the thread number two, what will be its length? It will be much larger than the thread number one, right? So in the same way, if there is a folding in case of a plasma membrane, if we just stretch it, what happens? It surely increases the surface area. That's what we meant by this. That's how these all these structures increases the surface area. Clear with that concept? So chromatophores, and what are these chromatophores? These are the pigment containing membranous infoldings or in some prokaryote. Example, in case of cyanobacteria. In case of cyanobacteria, we have cyanobacteria, also known as what? Blue-green algae, right? Blue-green algae. So In case of the cyanobacteria, we have this chromatophores. Okay, let me search how much we have to complete. We have to complete this much slides. I don't know whether we'll be finishing, but I will be moving very slowly, only making all the concepts clear. Okay with that? Okay, so it is formed of non-membranous circular genomic DNA, sing- single chromosome or genetic material and protein. Okay, so many bacteria have circular DNA or plasmid outside the genomic DNA. It gives some unique phenotypic characters to the bacteria. So I will explain this about the nucleoid. So, nucleoid is formed of non-membranous structures. Non-membranous naked. That means there is no outer covering. Circular, no, there is no outer circular covering for this genomic DNA. It is a single chromosome and it's their genetic material. Okay. Okay, fine. So, many bacteria have a small circular DNA or plasmid outside of this genomic DNA. Look, plasmid is also there. Out of plasmids are there out of this nucleoid, and it gives some unique phenotypic characters. That is the resistance to antibiotic is their unique phenotypic characters, right? Nothing more than that. Next is the another structure known as flagella. Next, another structure known as flagella. So these are the filamentous extensions from the cell wall of motile bacteria. Filamentous extensions, thin filamentous extensions. Their number and arrangement are varied from different bacteria. And bacterial flagellum has three parts: filament, hook, and basal body. Filament is the longest portion and extends from the cell outside, cell surface to outside. Okay, guys, is all the concepts clear till now? Is all the concepts clear till now? Fast till now, if everything is clear, it's okay and fine. Okay, so it has a motile flagella. It's the extension from the cell wall of motile bacteria. Their number and arrangement is varied in different bacteria, and it has three parts, that is the filament, hook, and basal body. And the filament is the longest portion and it extends from the cell surface to outside. So look, the structure that extends from the cell surface to outside is referred to as the filament. Clear? Pili and fimbriae. These are surface structures that have no role in motility. Pili are elongated tube-like structures made of special protein. Look, what is this? Look, these structures, the elongated ones, are known as the pili, and it is the sex piles that help in reproduction and not in motility. Fimbria are bacil-like fibers sprouting out of the cell. In some bacteria, they help to attach to the rocks in streams and to the host tissue. That such structures, these structures helps in the attachment to a host. We can say it as host, such as the host tissues of all other organs, and we say it as host, right? In concept of bacteria, it is host. So, for in further attachment, fimbriae are present. Next is the ribosome. So the type of ribosome present in this prokaryotes is 70s type. They are associated with plasma membrane of prokaryote. They are about 15 nanometer by 20 nanometer in size. They are made up of two subunits, 50s and 30s unit. They together constitute the 70s prokaryotic ribosomes. S refers to the sedimentation constant, so coefficient. Sedimentation means what? They will be accumulated in the top and top of that. So such a sedimentation coefficient is referred to as this S. So all the ribosome, prokaryotic ribosome is 70s ribosomes. 70 sedimentation coefficient ribosome. Then their size is 15 nanometer by 20 nanometer in size. All those are not required, but you should study the type of prokaryotic ribosome, that is the 70s, 70 sedimentation coefficient. That is a measure of density and size. So function of ribosome is the site of translation, that is the site of protein synthesis. Several ribosomes may attach to a single mRNA to form polyribosomes or polysome. So this is mRNA. So several ribosomes may attach to that and they form a chain known as polyribosomes or polysome. This is the direction of translation, direction of protein synthesis. Ribosomes of polysome translate the mRNA into proteins. That is what is said to be the protein synthesis. Next, we have the inclusion bodies. If everything clear till now, is everything clear and fine till now? I hope I was a little bit fast at the end. Do you want me to take it once again? Do you want me to explain it once again? Some construction work going on here. Yes, okay, clear, fine, absolutely fine. Next is the inclusion bodies. So what are these inclusion bodies? You can see in the diagram what are these inclusion bodies, right? So they are non-membranous and stored reserved material freely in the cytoplasm, just scattered in the cytoplasm. All these are phosphate granules, cyanophacine granules, glycogen granules, gas vacuoles. All these are the structures of this inclusion bodies. These are just stored reserved materials present in the cytoplasm of this prokaryotes. So gas vacuoles, what are these gas vacuoles? They are found in blue-green and purple-green photosynthetic bacteria. Okay, they are found in blue-green and purple-green photosynthetic bacteria. And that's all for the day. That's all for the topic of ribosomes. Clear? Everything clear and fine? Clear and fine till now? Yes, okay. So just read all the topics from the NCRT today itself, soon after the end of this session. Okay? And after that, after that, just practice questions. Roshida, questions for the NEET batch will be in the form of Telegram course and it will be did as soon as possible. Okay, per topics. Okay. These are just basics of the topics of prokaryotes and eukaryotes, which we did this much time, but all these are important. Right? Okay. And that's all for the day. Thank you for your patient listening. It's me signing out from your favorite and my favorite. Biopoint Stream, the world of bio. Bye bye.